In this video clip, let me introduce a friend of mine, Professor Ozawa from the University of Tokyo. Professor Ozawa is a leading expert of bioimaging. Welcome.
Nice to see you.
Okay, good to see you. Thanks for coming. I have a present for you. Here you go.
Thank you.
I have another one. Okay, here you go.
Thank you. Okay, I will give you a present box.
So this is something only video lectures can do, right? Okay, let's get back to the lecture. These 2 weeks, I told you about fluorescence. The Professor Ozawa and I would like to explain another kind of light emission, chemoluminescence. What is chemoluminescence?
Chemoluminescence is emission of light as a result of a chemical reaction.
So what is the difference from fluorescence?
Well, fluorescence is also called photoluminescence, which is a result of light absorption.
In the case of fluorescence, we need to shine the light to get the light back.
In other words, excitation light excites the molecule and the energy is released as emission light.
Right. In contrast, chemoluminescence does not require irradiation of light.
Yes, the energy comes from a chemical reaction. A particular chemical reaction generates a distinct molecule in an excited state, and the energy is released as emission light.
Do you have any good examples?
The best known example is luminol.
Oh, that's the one that detects blood.
Here is the chemical structure of the luminol.
How does that emit blue light?
This is the reaction.
Okay, let me explain the reaction. The first, luminol react with hydroxide anions and hydrogen peroxide to generate this excited molecule. This molecule emits blue light when it goes to the ground state. But I'm just wondering why luminol emits blue light in the presence of blood.
Blood has iron. Iron is a catalyst for this reaction. That's why luminol detects blood.
Any example from living organisms?
The best known example is firefly.
Here is a firefly right here. How does this happen?
Okay, let me show you how it works. Dr. Harvey of Princeton University has first discovered the luciferase reaction in 1922.
Luciferase is an enzyme, right?
Yes. Luciferase catalyzes the reaction that transforms D-luciferin to oxyluciferin in the presence of ATP and oxygen. The excited oxyluciferin emits photon whose emission maximum is around 560 nanometer.
So ATP is the original energy source. Let me remind you the structure of ATP.
ATP is considered as an energy currency of life. It is a high energy molecule that stores the energy.
Is ATP absolutely required for the reaction?
Yes. It's required. Let me show you something. The first analytical use of luciferase was achieved by Dr. McElroy of Johns Hopkins University long time ago. He demonstrated that concentration of ATP can be measured by luciferase.
The luminescence from luciferase is completely correlated with the concentration of ATP.
So he used the luciferin-luciferase reactions to measure ATP concentrations in crude samples.
In the next video clip, we will show you how luciferase can be used for biological analysis.